Thanks, Steve, and welcome all to the NOC for those of you that have not been here before. As Steve says, my name's Chris Horton and I'm Head of School for Ocean and Earth Science. And I'm really pleased to welcome you here, but also to introduce our first speaker, Stephanie Henson. Steph's an alumnus of Southampton, which is fantastic. She took an MSc in 2000 and finished a PhD in 2006. And then she's had a varied career. She's been over in the States at the University of Maine and then Princeton University in 2008, 2009. Came back to the UK as an NERC Research Fellow in 2010 and then has stayed in the NOC since. She's risen to the position of the Marine Biogeochemistry Group Leader. And in 2020, we were really pleased to nominate her for an honorary professorship at the University of Southampton. Along the way, Steph has been recognised for her talents and her research with a number of awards and highlights. So in 2012, she was awarded the European Geosciences Union Arne Richter Award. 2017, she was awarded a European Research Council Consolidator Grant. 2018, Challenger Society for Marine Science Fellowship Award. And in 2024, European Geosciences Union Fridtjof Nansen Medal. So clearly somebody of amazing caliber, and I think we're really fortunate to have people of this talent within the building and making contributions to the building, but also to ocean and earth science. So for a good number of years, Steph helped teach or delivered one of our modules for us in ocean and earth science, which we're hugely appreciative of. So I won't delay any further. Steph's going to talk to us this afternoon, down to the deep, the ocean's role in the carbon cycle, and tell us about some of her work understanding the deep ocean's control of global carbon. So I'll welcome Steph to the stage and hand over.
Thank you, Chris, for the really nice introduction. And when you read it all out in a long line like that, it does sound more impressive than it feels. Anyway, so hi everybody, I'm Steph Henson. I'm based here at NOC, but as Chris pointed out, I've held an honorary professorship with the university for about 4 or 5 years now. And my research all centres around the global carbon cycle, and in particular the role of biology in that. So just to get us all on the same page, I need to find the clicker, here it is. Just to get us all on the same page, I want to remind us about the importance of the ocean carbon sink in the global carbon cycle. So most of you probably know this already, but just as a reminder, the ocean is that great big blue part of the circle. About 90% of carbon on the planet is stored within the ocean. And what about the CO2 that we're releasing during our human activities, our anthropogenic activities? Well, a large proportion of it does actually end up in the ocean. So you can see over here, the atmosphere takes up a chunk, the ocean takes up about a quarter, and the rest is in the land. Now, these numbers have actually been revised recently, and the ocean contribution to the carbon sink has increased because they discovered errors in the methodology. So the ocean is now more important than the land for storing carbon in the latest estimates. But still, about half of anthropogenic emissions remain in the atmosphere. So as I said at the beginning, my work centres on the role of ocean biology in all of this. So where do the ocean organisms fit in? So this is one of those horrendograms which shows you all of the fluxes and stocks of carbon in the globe. And there's various things going here on the land, the atmosphere, and the ocean. And I just want to point out a couple of things. This is the pool of carbon stored in the ocean in organic forms. You can see it's by far the largest circle on there. So it's by far the largest stock of carbon in the globe. Marine biota is that tiny, you probably can't even see it. It's so small. It's that tiny, weeny little dot there. So that's the carbon stored in marine organisms, so in the bodies of marine organisms like whales and fish and everything else, plankton that lives in the ocean. And then this circle here and these arrows are meant to indicate that the cycling of organic carbon here between the marine biota and the organic carbon pool is static. So there's no net flux. You can see the numbers here, 80 in, 80 out. No net flux of anthropogenic carbon into the oceans due to biology. Now, this confuses people a bit because they think, well, you've just shown us that the ocean takes up masses of anthropogenic carbon. But actually, most of the anthropogenic carbon is taken up through the solubility pump, which is to do with the physics of the ocean. I'm not going to talk about that. I'm just going to talk about the biology part. So having just told you that it does nothing to reduce anthropogenic CO2 in the atmosphere, you're probably wondering why on earth have we got an entire group at NOC and elsewhere studying it if it's not doing anything. So I'll answer that question in a moment, but just to make sure we all understand how biology takes up carbon into the ocean and how it's stored, I'll just introduce you to one diagram of the biological carbon pump, as we call it. So at the sea surface, carbon dioxide is taken up by phytoplankton, which are tiny marine plants. Like all photosynthesizers, they're using up the CO2 as an energy source. They're then getting eaten through various complexities of the food web by larger organisms, zooplankton, bacteria, there's fish up here, and so on. And a tiny fraction of the primary production undertaken by the plankton up here in the top of the ocean sinks down into the deeper part of the ocean, what we call the twilight zone or the mesopelagic part of the ocean, which is from about 100 meters down to about 1,000 meters, something like that. So only about 1% of what's happening up here is actually sinking down below the surface. But generally speaking, the deeper that organic carbon can penetrate into the ocean before it's turned back into CO2, before it's remineralized again, then the greater the possibility of that carbon being sequestered, being stored on climatically relevant timescales, so timescales of hundreds of thousands of years. So that's what we're interested in, is that bit. So where does ocean biology fit in? So this is an ancient paper, but it's a really good one, and it's still valid today. So what this is showing us is that the vertical gradient in dissolved inorganic carbon in the ocean is primarily set up by the biological carbon pump. Around 2/3 of that vertical gradient is because of biology. So it's the difference, basically, between the B curve and the C curve is all of the biological contribution. But there's no net storage. As I just pointed out from that diagram earlier, there's no net storage of anthropogenic CO2. And that's because the biological carbon pump is, it seems, apparently, it's in steady state. So what it's drawing down is, in terms of CO2, is being replaced through circulation and remineralization by outgassing of CO2. That's the theory. And we'll see whether A, that's true, and B, how it might change in the future as we continue to pollute the atmosphere. So why do we care about the biological carbon pump? Okay, hopefully I'm starting to convince you. Now, the only way we can tell what happens if we don't have a biological carbon pump is through modeling. So in models, we can do fun things like just kill all the plankton, just kill everything in the ocean, kill all the life in the ocean. So what happens if you do that? Here you've got the control, which is like a normal, healthy ocean, and up here you've got a dead ocean where they've just killed off everything that lives in the ocean. And by doing that, you can see that atmospheric CO2 levels go up considerably, by about 150 to 200 parts per million. So if biology didn't exist in the ocean at all, then we'd have an even bigger problem on our hands in terms of atmospheric CO2 levels. Now, obviously, killing everything in the ocean, that's not realistic. Hopefully, that's not realistic, even in the most extreme futures. And so, although this gives us kind of like an upper bound, we can also see what happens in more realistic situations. And one of the ways that this has been done is through using paleo proxy data. So we're interested to know, okay, well, if the biological carbon pump's not really changing at the moment, as far as we can tell, has it been prompted to change in the past, in the paleo past? And the answer is it has. So down here is age in thousands of years, and we're looking here at the interglacial period. And what you can hopefully see is that during periods of when the Earth has been drier and the deserts have been greater in size, you get an increasing amount of dust being blown out of the deserts and deposited in the ocean. And that dust contains iron, which is a critical limiting nutrient for phytoplankton growth. And so here you can see there's a sudden increase in the amount of iron supplied to the ocean. And what you can hopefully see is in this green here is a proxy for primary production. And as the iron supply increases, so does the primary production. So the phytoplankton are really happy. This is good news. And what happens to atmospheric CO2 concentration in the purple line is that you get a massive decrease in atmospheric CO2 prompted by this change in the nutrient supply to the oceans. So the biological carbon pump has changed in response to different processes, different drivers in the past. And so we can ask the question, is it changing now? And this is a paper from earlier this year which gathered together loads and loads of data, in situ data, from all over the oceans and tried to make an assessment of whether the flux was changing or how efficient the transfer of organic carbon from the surface down to the deep ocean was. And basically, the data they compiled suggested that although flux out of the upper ocean here is increasing, mesopelagic fluxes are actually decreasing. And so you're seeing a decline in the efficiency of the biological carbon pump in the contemporary era, which would be worrying if it was true. I'm not going to slag the paper off too much, but let's just say I couldn't reproduce their results. So anyway, but so this is interesting. It's an interesting thought experiment that they've done this, and they said, okay, well, look, it looks like the biological carbon pump efficiency could be declining already. And how might it change in the future? Now, obviously, we have to turn to models to address how it's going to change in the future. And here's an ensemble of models from the IPCC Climate Modeling into Comparison project. And you can see this is the change in export flux, so the amount of organic carbon leaving the surface ocean over time as we go out into the 2100. And you can see for most models it's declining, some more than others. But the projected change isn't consistent between the models. It ranges quite massively. And in large proportions of the ocean, the models, and these are the best climate models we have, don't forget, the climate models disagree even on the sign of the change in carbon export, whether it's going to increase or decrease. So there's huge uncertainty in the modeled export flux and how it's going to respond to climate change. Now, I argue that there's a fundamental constraint that we have to understanding export flux and how it's transformed through the mesopelagic, because there's a huge number of interconnected processes that are happening in the twilight zone that make the biological carbon pump really hard to both observe and to model as well. I showed you a simple diagram of the biological carbon pump. Here's a more complex one. Don't try and read it. There's even more complex ones. Once you start delving into the biological carbon pump, it's just unbelievably complex. And there's all these different moving and interacting parts. And biology doesn't play ball in the same way that physics does. You can't write an equation for it necessarily in the same way as you can for a physical process. So the biological carbon pump is really hard to observe and to model. And one of the reasons that I think that we really struggle to know what's going on is the difficulty that we have in resolving how things are changing over time, so over seasons, over years, and over decades. Now we know, or we believe, that the biological carbon pump, its magnitude and its efficiency, should be changing over the course of a year, seasonally, because the different inputs change during the year. So, obviously, the amount of sunlight changes during the year, so the amount of primary production is changing through the year, so everything should be changing through a year. So it's unlikely that the biological carbon pump is just static all year round, but it's very hard to observe that variability. So not only is there bias in when and where we sample, but often the measurements we take are instantaneous. We take them on a ship, and we can only be out on a ship for certain limited periods of time. But, of course, we now have autonomous vehicles which are helping us to close some of those gaps. So autonomous vehicles can stay out at sea for months, years at a time, taking nearly continuous observations of things which are important for understanding the biological carbon pump. They can't capture flux, the carbon flux directly, but they measure things that we can use as proxies to get at it. So here's some examples. This is a glider, an Argo float, biochemical Argo float, and AutoSub, which is developed here at NOC in the building. So if we look at the kinds of data that you can get from that, this is from 4 months of a glider deployment near South Georgia in the Southern Ocean, where you can see the really high degree of temporal variability in the data. So the glider was returning to us in real time about 8 profiles per day down to 1,000 meters and back up. Now that's completely unheard of from a ship. There's no way you could do that, and especially not for 4 months. So for us, this was just amazing. We were like, wow, I got to look at all the detail that you can see. You know, if you imagine just taking a ship and just taking one sample here and another one a week later, another one a week later, you know, you just can't resolve this variability at all. So we're really excited to be able to pick out some details like particle plumes descending through the water column, for example. And so in an analysis we did of this data, I won't bore you with all the gory details, but basically what you can do is start to get really detailed time series over several months of how different aspects of the biological carbon pump are changing. So how the export flux is changing, how the efficiency of these things are changing. And we found really high temporal variability, not just on the seasonal timescale, but also sort of episodicity as well. So events coming in and out that we would just miss on a ship, or we would just all mischaracterize them if we tried to capture them from shipboard data. And carrying on, so that was the near the surface stuff, and carrying on down into the twilight zone, we found really quite large changes in the efficiency with which organic carbon is being transferred from near the surface ocean down to the deep ocean. And that's important because, of course, the deeper it gets, the longer it could potentially be stored for out of the atmosphere. So we found these just big swings, both on the seasonal timescales, but also at daily, almost, time steps as well. So this was really eye-opening because we hadn't had data at this resolution before. And, well, I don't know, I kind of guessed that there would be this much variability in it, but to be able to quantify it was really fantastic. And so what might be causing some of this, you know, variability that we're seeing? And if you delve into the literature, most of the time people think that the remineralization is going to be driven by temperature, because we know that when temperatures increase, metabolic rates increase as well. Everything goes faster. That works for humans and it works for bacteria, which are breaking this organic carbon down into inorganic carbon. However, during the course of this time series, the temperature barely changed at all. It just really didn't change. The temperature didn't change, the oxygen concentration didn't really change, but you see this huge variability in what we're predicting the flux attenuation to be, which was actually larger at one location over 4 months than the entire predicted global range. So it's massive variability, and you know, the temperature and oxygen data did not back up at all, being main drivers of that variability. So the conclusion we came to was that it must have something to do with the community structure. So the structure of the phytoplankton and zooplankton communities, the diversity basically of organisms in the surface ocean and in the mesopelagic as well. So uncovering that whetted our appetite to go and look further into this, which brings me to a new, well, it's ongoing, it's still happening, this project is still going, called Party Tricks, which is part of the biocarbon program funded by NERC. And I didn't come up with this acronym, but I like it. So Party Tricks is about the transformation and respiration of organic carbon and how it influences carbon storage in the ocean. And our goal was basically to work out what is the interaction between the biota and the particles, and how that's changing with depth and season, and what the consequences of all of that are for the ocean carbon storage. And our goal was to try and generate information from the data that could be used for modeling, specifically. So not just to fill in the gaps, but generate data used for modeling. So this is what we did. So this is from the proposal. Believe it or not, all of those instruments and autonomous vehicles and things dangling from ships and processors, we managed to get information about all of them in one 6-week cruise while sharing the ship with 2 other projects. So it was exhausting. So the floats are still out there right now. Once we deploy them, we don't recover them anymore. So those biogeochemical Argo floats are still returning data. Everything else was recovered over the course of the cruise, or there was a subsequent cruise as well where we collected some stuff. And the kinds of things we're interested in is the migration up and down each day by zooplankton. We were interested in how particles stick together and how they break apart, because we know that's important. We were interested in how particles get respired by microbes or by zooplankton. And we're interested, of course, in things sinking down. That's the main way that particles make it to the ocean, into the deep ocean, is just sinking. So we had ways and means to measure all of this, in theory. So we went to sea in June last year, and we deployed the floats, and then we tried to follow them around. Because, of course, with the ocean being an ever-moving substance, it's difficult to disentangle the spatial variability and the temporal variability from each other. So we tried to follow the floats around, which was semi-successful. And we deployed a million things, and I'm not going to tell you what all of these are. All you need to know is we generated an absolute mountain of data, and it's going to take us a decade to wade through it all, frankly. It's going to keep students in business for years. Right. So this is a little movie which shows where they all went and what they all did. So some of these are gliders, you can see up here in the sort of yellowy colours, and some of them are floats, which are in the blue colours. And they all went whizzing around, and the ship tried to chase after them. I want to highlight the work of Philippa and friends, who are now helping to design. This is for the tech geeks out there. So we get all of this information. I mean, we get masses of data back from our autonomous platforms, not just from this cruise, but from other ones we've got done. We get masses. And in the past, we've always done the QC calibration and the data processing bespoke for each deployment, which just massively slowed us down. And so now Philippa and co are designing an automated pipeline, which is going to revolutionize how we deal with this data, because we'll be able to get to science-ready data way, way quicker than we currently can. Because currently it takes us years. I mean, that's not an exaggeration, it takes ages. So the hope is that this will speed everything up and we can get writing those cool science papers quicker. So this is the data that we collected from the Iceland Basin mission. And so again, you can see just the richness of the data here. This is actually chlorophyll, I think is that what it says? No, temperature. This is actually temperature, but we have this for chlorophyll and particle concentration and so on as well. And again, just very, very rich data set, very high temporal resolution. And I'm just going to have a little, very, very brief, quick whiz through some of the cool stuff that we've been doing with this data, just to sort of, I mean, I'm not going to dwell on anything because it's too much of it, but pictures. So these are images taken from an instrument called the Underwater Vision Profiler, and this is one of our favorite tools. So it's a really amazing imaging device that we can deploy on various platforms, autonomous and shipboard, and it takes, as you can see, photographs of the stuff in the water. So these are some beautiful images that Amanda's pulled out. Actually, she sent me some this afternoon, which were even more amazing, and I couldn't put them because I had to upload this talk earlier in the week, but I wish I could have shown you them. So you can just see how fabulous they are, and we've been working on automated classifiers so that we don't have to manually go through millions, and it is millions and millions of images each time we deploy one of these instruments. So we're working on AI-driven classifiers so that we can speed all of that up. And that's really successful. That's in collaboration with our colleagues in Laboratoire Villefranche. Something that Nathan has been working on is looking at drivers of particle aggregation, so how they all stick together. And he's been exploiting, again, the very rich data that we get from the autonomous platforms. So in this case, he's looking at float data. And what that UVP imaging system I showed you those beautiful photos from earlier can also do is tell you about the size of the particles in the water column. And that's really important because, of course, if particles are sticking together, they're getting bigger. If they're being broken apart, fragmented, they're getting smaller. And as they're remineralized and turned back into inorganic carbon, they are also getting smaller. So the size of particles is actually super useful information for us to understand the processes going on. So what Nathan's been doing is looking at different layers in the ocean, the upper layer, middle, and deeper down, and just looking at how the particle size distribution is changing over time. And so right at the beginning of the bloom, we see loads and loads of small particles, but we do see a few making it up to the bigger sizes. So turbulence is able to aggregate some of the small particles, lots of small particles, into some bigger particles in the surface layer. But when turbulence becomes lower, as you go deeper down into the water column, the aggregates actually manage to get larger. So the size distribution shifts towards larger particles. And if you get a bit further down, then we see pulses of really quite large particles reaching down into the mesopelagic. So the size structure changes a lot during the course of the seasons and also with depth as well. And what Nathan's doing now is incorporating turbulence data and TEP, which is a stickiness factor that we can measure in the ocean with great difficulty. So he wants to try and model these aggregation dynamics. And this is being helped out by Chelsea, who actually took the TEP measurements at sea, and she found some really enormous aggregates. So this is a bit of particle detritus. It's a bit of fluff. And you can see there's a 2 millimeter scale bar here. So this thing is huge. You know, when I'm talking about small particles in the ocean, I'm talking about things which are, 100 microns or less, 20 microns. And this is 2 millimeters, just that. So this is absolutely enormous. It's like the size of one of those dust balls you find behind your couch, and about the same sort of shape and structure. It's basically a bunch of stuff that's got stuck together. But it's full of carbon. It's full of organic carbon. So we find these really large aggregates in some places. And the particle imaging shows us that we find these extremely large particles just below the mixed layer. So at the same place where Chelsea finds a peak in stickiness and turbulence, she finds these massive, massive aggregates, which suggests that something is causing them to stick together, the turbulence is bringing them together, and the stickiness, whatever, we're not quite sure what the stickiness is, the stickiness is helping them to form these massive aggregates, which I can only imagine will probably sink relatively quickly into the ocean and carry lots of organic carbon with them. So next, she's working on combining these different data sets to model the aggregation dynamics. So further work that Nathan's is doing is looking at particle fragmentation. And this was a real surprise to us. Every time we go to sea, we see something surprising. But this was really cool. So on the CTD, which is the main sort of frame that we use to take samples, it's a great big round thing. It's probably a couple of meters in diameter. And we deploy it over the side of the ship and then pull it back up. It takes water samples. And we had one of these camera systems on the CTD frame. And what we found was that when we go down the water column, you see quite a lot of aggregates. That's what all these spiky bits are in the data here. But when you pull the CTD back up through the water, they're all gone. That's the blue line. And so we were like, what? That was only an hour ago, and now they've all gone. Well, no, actually what's happened is that they've been broken down, probably just by the turbulence caused by the CTD frame being pulled back up through the water by the ship. And as you go from the early part of the cruise to the late part of the cruise, you can see that the difference between these two, the pink and the blue lines, increases, which means there are more fragile particles available to be broken apart in the later part of the cruise. We've got no idea why, but it's fascinating. So we're going to be digging into that a bit. And just to say that we also collected actual samples. A lot of that has been based on sensors and imaging systems, but we did also collect actual physical samples as well of what was in the water column and the sinking particles. And Elena's been doing this work, looking at the fraction of different organisms and detrital types that she found from her samples. And basically, we get, differences over the time period and differences in the depth as well. And she's going to be busy relating the differences in particle composition to how quickly they're respired. And many other data sets, and I don't want to go into them because we'll be here all night, but we collected data on zooplankton abundance, microbial respiration. We got some additional funding to process the auto-sub data and integrate it with other data streams. And there's a really big focus in Party Tricks, as I think I mentioned at the beginning, about synthesizing our data together with other data sets to try and scale up our results beyond the one cruise that we had. Now, why are we interested in doing that? We know we've obviously got lots of fascinating stuff that's going to last us decades just in the cruise data. But we want to make this a big part of our project because we want to generate data by scaling up that are on the right space and time scales for model development and model validation. So why are we bothering with that? Why do we want to do that? And I mentioned at the beginning of the talk that the biological carbon pump is difficult, really difficult, to observe adequately. So this is that same data set I was showing you before from the model output from the IPCC's Model Intercomparison project. It's just plotted in a different way. So this is the absolute export flux out to 2100. So you can see that decline that's predicted in most of the models. Now, you might say, okay, there's a huge range in the models. Surely we can decide which of those models is more robust than another one by looking at the observations, and the observations will give us some kind of constraints on which of those models is a good one and which one is a bit not so good. Well, the model range in global export in the pre-industrial era is between about 5 and 12 gigatons of carbon per year. Well, unfortunately, the range in the observations-based estimates of global export is 5 to 12 gigatons of carbon per year. In other words, it's exactly the same as the model estimates. So the observations provide absolutely no constraint on which of these models is more robust than the next one. So that is a massive problem. We can't even use the data we have to try and narrow down which of these is a good model. We have problems scaling up. That's what the problem is. So we can go out and make all sorts of fabulous measurements from ships, but they're just here, there, this time of year, that time of year, almost impossible to synthesize together. But luckily, we do have new technology, holds promise, which is these autonomous vehicles. So, we have this issue of data sparsity. This is the location of all of the shipboard particle flux observations from 1975 to 2025 that someone has managed to gather together. And there's a lot of data here, but there's huge gaps. I mean, there's areas of the ocean which have never been sampled at all for carbon flux. And don't forget that these data points represent a single point in time. They don't tell you anything about the seasonal variability either, or the interannual variability. So this is all that we have at the moment from shipboard data. However, you can compare that to the location of biogeochemical Argo floats from the last 12 years. And you can see that although there's areas of the ocean which still aren't sampled, there is much, much greater data density. And these floats, of course, are giving us information that's temporally resolved as well, so we can think about the seasonal and interannual variability. The downside, of course, is that these profiles, this data, these sensors don't give us actual flux measurements. They give us proxies, but they are nevertheless extremely, extremely useful and hold great promise. So if shipboard data is really very sparse, which we can see there that it is, what's the influence of that on our best guess of what's happening with the biological carbon pump? So we can take a data synthesis like this, which was published a few years before Simon's. And you can see data sparsity, very clumpy as well, because we go out on a ship and we tend to do transects. We don't really cover large parts of the ocean. So what effect does that have? We can try and answer that question by subsampling a global biogeochemical model at the same times and places as we have the observations. So there's the black dots on top, that's the observations. Underneath is a model, so we can just pull out the data at those places. And then we can try and reconstruct the full field of the observations from those subsampled points and find out what happens. How good is that as an estimate? So we reconstruct the field using this linear relationship between primary production and export flux. And that is the difference in the reconstructed flux from the true model flux. And you can see that the values here are pretty big. Quite a large proportion. In fact, we find that it's higher, this estimate is higher by about 20% than the true model flux. So that's because the data is so clumpy and so sparse, we ends up just completely overestimating the actual carbon flux. Now, we could say, well, that's just because of this particular arrangement of the data. So we can make some synthetic data sets. We can subsample the model using the same statistical characteristics of this data set in terms of how many data points there are and how close they are together and where they're sampled. And we can create a bunch of synthetic samples. So here's another fake data synthesis, which has the same statistical characteristics as the real one, and another one, and we can keep subsampling the model over and over again with these similar sort of patterns and build up a distribution of what's happening. And there it is. So the integrated total global carbon export, the histogram you see there, it ranges between about 8 and 12 petagrams of carbon per year, which is still very large range. So that's not really helping. It does suggest that our in situ data syntheses are biasing our understanding. However, of course, we know from the biogeochemical Argo data set that I just showed you, we have much, much more data there. So what happens if we subsample a model with this pattern instead, which is the biogeochemical Argo backscatter profiles? And I've done that for every year. And what you can see is once it settles down and the number of profiles being collected gets to a certain amount, it settles down and the variability is hovering around 10-ish, and it's just varying gently around that by about half petagram of carbon per year. So it's a much more constrained, lower uncertainty estimate of the global export flux. So that's good news, a much tighter constraint. So the Argo, BGC Argo network does have the potential to provide tighter constraints on export flux. But I just want to point out that this interannual variability in the export that we estimate, I'm subsampling the same model year all the time, so the model is not varying year to year. This variability year to year in these estimates is simply due to the changes in the sampling pattern of the BGC Argo float network. So just that year-to-year variability in where the floats are deployed and where they're moving around generates already quite a large amount of uncertainty. And that uncertainty is actually larger than the climate change-driven trend in most of these models. So we may still yet have a problem in detecting a climate change trend in export flux robustly, even with the BGC Argo network of profiles. So how long have I got left, Chris? What am I supposed to finish? 5 minutes, okay. I'm going to take a very brief detour into something which has been tickling my brain, and when I say brief, it's like 2 or 3 slides, about what role small particles might play. Now, typically, we assume that most of the action in the biological carbon pump is done by large particles because they sink quickly, and large particles do carry most of the organic carbon flux. But we do find an enormous quantity of small particles in the ocean as well. And backscatter sensors on autonomous platforms like Argo floats capture this small particle signal really, really well. And the question is, if those small particles are being undersampled by our usual shipboard measuring devices like sediment traps, does that mean that we're missing something if we're only concentrating on the large particles and we're just ignoring the small particles? So James Williams, one of our PhD students, has been looking at the BGC Argo network, the profiles, and what he's been finding is this, to us, very strange-looking profile. So typically what would happen is organic carbon in the ocean should decline quite smoothly with this sort of shape. And instead, what he's finding is that although there's an initial decline, then you get this massive blob of organic carbon just sitting there in the middle of the twilight zone, in the middle of the mesopelagic. It should be being remineralized, but it's not. It's just sort of sitting there. And this is the frequency with which he finds these blobs in the ocean. And he finds that about 1 in 5 profiles globally have got one of these bumps in them. And in some regions, it's much more prevalent than that. So he's digging into that. We also have been using a data set from our colleagues in Villefranche again, where we've been looking at small particle losses in the mesopelagic. And we find that small particle loss is greater in high latitudes than in low latitudes, which was completely the opposite to what we're expecting. So what we've got over here is the temperature and this transfer efficiency in the small particles. The transfer efficiency is the fraction of small particles remaining at 1,000 meters versus what was leaving the surface ocean. I said earlier that temperature controls remineralization rates. When it's warmer, you expect more remineralization, which means that when it's warmer, you expect fewer particles to survive their transit through the water column. So this should have a negative slope. In fact, we find the exact opposite pattern. I've got no idea why, but there it is. But we've been turning to models to help us to try to answer this question. So we've got two versions of the same model here. This is a model from, I haven't put it there, it's from Olivier Aumont, it's the Piskies model. And this one has no variable particle lability. The particles are all remineralized at the same rate regardless. Whereas this one has a variable particle lability, so there's some variability in how easy it is for the organisms to break down those particles. And the observations are down here. And you can see the pattern matches much better. So we think there's a role of particle lability here as well. Okay, just to finish up then and summarize where we are with all of this. So I hope I've convinced you that the biological carbon pump is important and really interesting, but like stupidly complicated. And we still don't know how to model it very well. And that leads to the situation, I'll just bring all these up, where we don't know what the feedbacks are going to be with climate change. So this is a paper we published a little while ago where we sat down and thought about all of the different feedbacks that might exist to changes in different processes in the biological carbon pump and whether they would generate a positive feedback, a negative feedback, or we don't really know. We can't put a number on that one. And I just want to highlight here the role of temperature. So we know that temperatures are going to warm in the ocean. That's pretty obvious. We've got that one down. Temperatures are going to warm, so we're going to get faster remineralization, faster grazing, which should generate a positive feedback. Over here, we've got temperature. It's going to warm, but that's going to decrease water viscosity and density, which will increase particle sinking speed. That's going to generate a negative feedback. And over here, we've got temperature, which is going to warm, and it's going to change vertical migration depth, and it's going to generate an uncertain feedback. So even something apparently as blindingly obvious as the water is going to warm, we don't know which of these three options it could generate in the biological carbon pump as a response, and therefore what the direction of those feedbacks could be. So we're really stuck, is a short answer. So remind me again why I care. It's getting late. You're like, oh, I've been talking about biology for ages. Right, so the reason we care is, so this is the latest global carbon budget. These guys from Exeter do an amazing job every year of collating all of the information out there about where carbon is going in the ocean. And the models are here, the data is here, and this gap between the models and the data has not really changed for the last umpteen years that they've been doing this. And I've been saying for a while that I think that this is partly due to poorly represented biological processes in the models. And I was greatly cheered to read, in this paper which just came out last week in Nature, that in fact they highlight in their conclusions that one of the areas needing better understanding is variability of the biological carbon pump. So I was like, yes, someone's finally listening. Okay, so that's good. And just to conclude then, it's not just me banging on about it, although I did write this. So maybe it is just me banging on about it. But in the IPCC assessment report, the last assessment report, in chapter 5, which is about carbon and biogeochemical cycles, we wrote that there was high confidence that climate change is going to result in alterations to the magnitude and the efficiency of the biological carbon pump, but we have low confidence in the magnitude or even the sign of these feedbacks. So there is masses of work that needs to be done just in trying to understand all of this stuff. So I hope I've convinced you the biological carbon pump is an important sink for CO2, but at the moment probably not for anthropogenic CO2, but that could change because it's changed in the geological past. Who knows if it's changing now? And the model studies at least suggest it will change into the future. And modelling this response and the feedbacks is hampered by the sheer complexity of what's going on in the twilight zone and the sparsity of observations that we have. And this might sound like a stupid thing to say, but I'm going to say it anyway because we're not all oceanographers here. But the ocean is really big. It's really big, but it's also really deep. So the volume to be sampled is absolutely massive, and most of it is completely inaccessible. We just can't sample it. So we really are hampered just by not being able to get out there, either with autonomous vehicles, with ships, satellites, whatever it is. We just can't sample that twilight zone well enough. But, the interplay between diversity, climate change and carbon fluxes is really an emerging research area, and there's loads of cool stuff ahead. So I'm looking forward to that. And thank you.
Yeah, thank you, Steph, for a fascinating talk and sort of the complexity of modern oceanography. Really fascinating. We've got time for some questions, so are there any questions in the audience? Okay, question there.
Thank you. Hang on. Can you just wait? Thank you. Hello. I was just wondering, at the very end with the small particles, when you measure the amount of small particles, how do you differentiate what is POC and what is PIC?
Oh, yeah, so PIC is particular inorganic carbon, for those who don't know, and we want to try and separate it from the POC, which is a particular organic carbon. And in this particular case, we haven't done that yet, but with the BGC Argo flows, you can look at the ratio between the chlorophyll and the backscatter sensors, which should tell you some information about if you're seeing very high levels of PIC, that should be reflected in there. But this is not, we haven't done it in detail for that example, but we know that some of those huge peaks in like the Arabian Sea, for example, are not due to that change in PIC to POC ratio. Yeah.
Any other questions? Justin. Microphone.
Just wondering, with your analysis of the sampling and how that affects your ability to kind of scale up and compare with the models, is there a way that you can optimize where you're sampling to get the kind of biggest bang for the buck in terms of knowing what's happening at the larger scale and improving models?
That's an interesting point. So one way that you could do that, for example, is to look at where there's greatest disagreement in the models and try and target your observations to that zone to try and narrow down the uncertainties in the models in those regions. The autonomous vehicles, so in theory you could do that with a ship as long as it's not like really in the middle of the Pacific where you struggle logistically to get a ship to. And in theory you could do that with the autonomous floats as well, but you still have to deploy them. So unless you've got a ship that's passing through the nowheresville of the South Pacific for some reason, and can deploy an autonomous float for you, it's really hard to get into those zones. So I like the idea of like trying to target regions of highest uncertainty, but we have such logistical constraints on the ships in particular that it's not always possible to do that.
Any other questions? I've got a question then to wrap up. And perhaps a stupid question. Is there any way that you can replicate what you're seeing sort of with that flux in the lab? Can you conduct experiments in the lab to give you insights into the processes that are happening in the ocean that you can't see all the time?
A bit, some processes. So some labs are equipped with roller tanks. So you create a particle and you get the tank rolling at a certain speed so the particle is sort of suspended because the water's always moving. So it's like the particle is sinking, but it's actually suspended, so you can keep looking at it. And the kinds of experiments that people have been able to do with that are around things like aggregation and fragmentation, where you can control the turbulence levels, for example, within those roller tanks. So you can look at things like that. What's more difficult is if you want to introduce any kind of organism in there to see how, for example, zooplankton might interact with the particles to eat them or to break them apart. And I don't know if I've seen any work that does that. There have also been some really cool pieces of analysis done with, I don't even know what the word is for them, but some very, very high resolution systems which have developed not for particle imaging at all, where they can suspend a single particle in a flow field so that its sinking speed is balanced exactly by the flow of the material upwards, the water upwards. And then they have ways of doing extremely high velocimetry imaging to see the dynamics of the fluid flow around the particles. And that's really, really cool. And they've even built systems which can go on a ship, so they're gimbaled. So in theory, they should stay steady on the ship, so you can do these kind of experiments even at sea, which would be really cool.
One final question over here, I think.
Thank you. Very interesting, but I'm a bit stuck on this perhaps word. So this is totally a different field for me. So perhaps, what does that mean for this, the need that we hear about to reduce anthropogenic CO2?
Oh, well, yeah, obviously we still need to reduce anthropogenic CO2 because there is only so much the ocean can take. The ocean takes up huge amounts of anthropogenic CO2, but mostly that's done through the physical processes in the ocean. And that's just to do with the amount of CO2 that you can, the solubility, basically, of CO2 in seawater. So most of the anthropogenic CO2 uptake is done through that process. However, what I showed before was that the background CO2 uptake and storage of the ocean, if you switch off the biology, you get this massive increase in atmospheric CO2. So it is playing its role. It's just that at the moment we think it's in steady state. So what is being absorbed and stored by biology is being re-exchanged again. And I say perhaps because we know it's changed in the past, in the paleo past, and the models predict that it will change in the future. And our observations are probably not really good enough right now to tell us if it's responding to climate change right now, but maybe it is. And we need to know about it, because if it is, that's going to be an extra problem in terms of the ocean taking up anthropogenic, potentially the ocean sink reducing even further, basically.
Okay, I think if we can just show our appreciation to Steph one more time for a really fascinating talk.